Now we will read the chapter biological classification. Okay, let's start. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. Aristotle used simple morphological characters. Okay. He also divided animals into two groups. Aristotle also divided animals into two groups: those which had red blood and those that did not. मतलब उनको एनिमल्स को दो ग्रुप में डिवाइड किए थे एरिस्टोटल, जिनका रेड ब्लड होता था और एक अदर टाइप का जिनमें नहीं होता है. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdoms was developed that included all plants and animals respectively. This system was used till very recently. This system did not distinguish between the यूकारियोट्स एंड प्रोकारियोट्स. ये एक ड्रॉबैक था लिनियस का, जो यूकारियोट और प्रोकारियोट में डिफ्रेंशिएट नहीं किया था. Unicellular and multicellular organisms and photosynthetic green plant and non-photosynthetic organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into this category. Hence, the two-kingdom classification used for a long time was found inadequate. A need was also felt for including, besides gross morphology, other characteristics like cell structure, nature of cell, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification systems for the living organisms have hence undergone several changes over time. Though plant and animal kingdoms have been a constant under all different systems, the understanding of what groups or organisms we included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over time. Characteristics of the five kingdoms. Characters five kingdom: Monera, Protista, Fungi, Plantae, Animalia. फाइव किंगडम्स में कौन कौन से ग्रुप हैं? मोनेरा, प्रोटिस्टा, फंजी, प्लांटी, एनिमिलिया. सेल टाइप क्या है? मोनेरा का प्रोकैरियोटिक, प्रोटिस्टा यूकैरियोटिक, फंजी यूकैरियोटिक, प्लांटी यूकैरियोटिक, एनिमिलिया यूकैरियोटिक. सिर्फ मोनेरा प्रोकैरियोटिक है. सेल वाल मोनेरा का नॉन सेलुलोजिक, ऑब्वियसली प्रोकैरियोटिक है. पॉलीसैकेराइड प्लस एमिनो एसिड मिलके होता है. प्रोटिस्टा प्रेजेंट इन सम, सेल वाल कुछ में होता है, कुछ में नहीं होता है. फंजी ग्रुप में सेल वाल प्रेजेंट होता है, विथआउट सेल्यूलोज होता है, सेल्यूलोज नहीं होता है. प्लांटी में प्रेजेंट सेल्यूलोजिक होता है, एनिमिलिया में एब्सेंट होता है. देन न्यूक्लियर मेम्ब्रेन मोनेरा में एब्सेंट, बाकी सब में प्रेजेंट. बॉडी ऑर्गेनाइजेशन मोनेरा प्रोटिस्टा में सेल्यूलर होता है. फंजी में मल्टीसेलुलर और लूज टिश्यू, प्लांटी में टिश्यू और ऑर्गन, एनिमिलिया में अच्छे से पूरा टिश्यू, ऑर्गन सिस्टम सब होता है. मोड ऑफ न्यूट्रिशन मोनेरा का और प्रोटिस्टा का ऑटोट्रोपिक है. मोनेरा, प्रोटिस्टा एंड प्लांटी भी ऑटोट्रोपिक है, लेकिन डिफरेंस क्या है? ऑटोट्रोपिक होता है मोनेरा में, ओके, केमोसिंथेटिक एंड फोटोसिंथेटिक एंड हेटेरोट्रोपिक, सैप्रोफाइटिक में भी पैरासाइटिक, जिस किसी हिसाब से वो खाना लेता है खाने के लिए. प्रोटिस्टा का ऑटोट्रोपिक, फोटोसिंथेटिक होता है वो, एंड हेटेरोट्रोपिक भी हो सकता है. फंजी में हेटेरोट्रोपिक होता है, सैप्रोफाइटिक और पैरासाइटिक. प्लांटी में ऑटोट्रोपिक, फोटोसिंथेटिक. और एनिमिलिया में टोटल हेटेरोट्रोपिक, होलोजोइक हो सकता है, सैप्रोफाइटिक हो सकता है. ठीक है. नेक्स्ट विल स्टडी about R.H. Whittaker. R.H. Whittaker 1969 proposed a 5 kingdom system of classification. ये जो क्लासिफिकेशन हम पढ़े ये किसने दिया था? R.H. Whittaker. The kingdoms defined by him were named Monera, Protista, Honji, Planti and Animalia. The main criteria for classification used by him include cell structure, thallus organization, mode of nutrition, reproduction and phylogenetic relationships. टेबल 2.1 जो हम लोग पढ़ चुका है gives a comparative account of different characteristics of 5 kingdoms. Let us look at this 5 kingdom classification to understand the issues and considerations that influenced the classification system. Earlier classification systems include bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms and the angiosperms under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which wildly differed in other characteristics. It brought together the prokaryotic bacteria and blue-green algae with other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones. Say, for example, Chlamydomonas, Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants, though they also showed a characteristic difference in their walls and composition. The fungi had chitin. Fungi के cell wall में chitin होता है in their walls, while the green plants had cellulose. Green plant के wall में cellulose होता है. Next, cell wall. Then, when such characteristics were considered, the fungi were placed in a separate kingdom. Kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera. Prokaryotic सब कहाँ रहते हैं? Kingdom Monera के अंदर. And the unicellular eukaryotic organisms were placed in the kingdom Protista. Kingdom Protista has brought together Chlamydomonas, Chlorella, earlier placed in algae within plants and both having cell walls, with the Paramecium and Amoeba, which were earlier placed in the animal kingdom which lack cell wall. It has put together organisms which in earlier classification were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological and reproductive similarities but is also phylogenetic, that is based on evolutionary relationships. In this chapter, we will study characteristics of kingdoms Monera, Protista and fungi of the Whittaker system of classification. The kingdom Plantae and Animalia, commonly referred to as plant and animal kingdoms respectively, will be dealt with separately in chapter 3 and 4. We will still study about that the later chapters. Okay, now we will study about kingdom Monera. Bacteria are the sole members of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under 4 categories based on their shape. The spherical coccus bacteria, this one spherical coccus bacteria, cocci, the rod-shaped bacillus, ये है rod-shaped bacillus, the comma-shaped vibrio, and the spiral स्पाइरिलम, ये स्पाइरिलम है, ये विब्रियम है, ठीक है। तो ये है कोकी बैक्टीरिया, बेसिली, स्पाइरिला एंड ये विब्रियो। 4 तरह के बैक्टीरिया होते हैं। Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic, autotrophic, or chemosynthetic autotrophic. So they may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs. That is, they don't synthesize their own food but depend on other organisms or on dead organic matter for food. Now we will read about Archaebacteria first. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas, halopines, hot springs, thermoacidophils, and marshy areas, methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows, buffaloes, and they are responsible for the production of methane biogas from the dung of these animals. Now we will study about eubacteria. Eubacteria. There are thousands of different eubacteria or true bacteria. Eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have chlorophyll A similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial or filamentous marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often formed blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts, that is nostoc and anomena. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are the most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in a legume, roots, etc. Some are pathogens causing damage to human beings, crops, farm animals, and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Sometimes under unfavorable conditions, they produce spores. They also reproduce by a sort of sexual reproductions by adopting a primitive type of DNA transfer from one bacterium to another. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Here two pictures are given. Let us see it. ए फिलामेंटस ब्लू ग्रीन आल्जी नॉस्टक, ये नॉस्टक का एक पिक्चर दिया गया है। ये हेट्रोसिस्ट फॉर्मेशन हुआ है और बाहर का जो कवरिंग शीट है उसको म्यूसिलाजिनस शीट कहा जाता है। फिर और एक पिक्चर है ए डिवाइडिंग बैक्टीरियम। ये बैक्टीरिया है जो डिवाइड कर रहा है, डीएनए डिवाइड हो गया है, सेल मेम्ब्रेन डिवाइड हो गया है, अंदर से फ्यूरल फॉर्मेशन हो रहा है, साइड साइड से श्रिंक होके और सेल वॉल बन गया है। ठीक है, अभी हम देखेंगे किंगडम प्रोटिस्टा के बारे में। स्टोरी नाउ किंगडम प्रोटिस्टा। A single-celled eukaryotes are placed under protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protista to one biologist may be a plant or another plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans under protista. Members of protista are primarily aquatic. These members of protista are primarily aquatic. This kingdom forms a link with the others dealing with plants, animals, and fungi. Being eukaryotes, the protista cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually by a process involving cell fusion and zygote formation. Now we will study about chrysophytes. This group includes diatoms and golden algae. Chrysophytes include diatom and golden algae, desmides. They are found in fresh water as well as in marine environments. They are microscopic and float passively in water currents, plankton. Most of them are photosynthetic. In diatoms, the cell walls form two thin overlapping cells which fit together as in a soap box. The walls are embedded with silica and thus the walls are indestructible. The diatoms have left behind large amount of cell wall deposits in their habit. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtering of oils and syrups. Diatoms are the chief producers in the oceans. Now we'll see about dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on the main pigments present in their cells. The cell wall has stiff cellulose plates on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the wall plates. Very often red dinoflagellates goniolates undergo such rapid multiplication that they make the sea appear red. Toxins released by such large numbers may even kill other marine animals such as fishes. Now we'll study about euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle, which makes their body flexible. They have two flagella, a short and a long. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they become like heterotrophs by predating on other smaller organisms. Interestingly, the pigments of euglenoids are identical to those present in higher plants, that is euglena. Slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves, engulfing organic matter. Under suitable conditions, they form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms fruiting bodies bearing spores at these tips. The spores possess true walls. They are extremely resistant and survive for many years, even under adverse conditions. The spores are disappeared by air currents. Protozoans will study now. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in fresh water, seawater, or moist soil. They move and capture their prey by putting out pseudopodia false feet as in amoeba. Marine forms have silica cells on their surface. Some of them, such as entamoeba, are parasites. Flagellated protozoans. The membranes of this group are either free-living or parasitic. They have flagella. The parasitic forms cause diseases such as sleeping sickness. For example, trypanosoma, ciliated protozoans. These are aquatic, actively moving organisms. Because of the presence of thousands of silica, they have a cavity gullet that opens to the outside of the cell surface. The coordinated movement of rows of cilia causes the water laden with food to be streamed into the gullet. Example, paramecium. Then sporozoans. This includes diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, malarial parasite, which causes malaria, which has a staggering effect on human population. Then kingdom fungi. Let's learn about kingdom fungi. The fungi constitute a unique kingdom. 1 minute. Sporozoans left, na? Okay, let's study about sporozoans. This include diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, malarial parasite, which causes malaria, which has a staggering effect on human population. Kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. When your bread develops a mold or your orange rots, it is because of fungi. The common mushroom you eat and toadstools are also fungi, while white spots seen on mustard leaves are due to the parasitic fungus. Some unicellular fungi, that is yeast, are used to make bread and beer. Other fungi cause diseases in plants and animals. Wheat rust causing Coccinia is an important example. Some are the source of antibiotics, that is penicillium. Penicillium is a source of antibiotics. Fungi are cosmopolitan and occur in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infections. With the exception of yeast, which are unicellular, fungi are filamentous. Their bodies consist of long, slender, thread-like structures called hyphae. The called hyphae, इसको hyphae बोला जाता है. The network of hyphae is known as mycelium. Mycelium किसे कहा जाता है? Network of hyphae को. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called कोइनोसाइटिक hyphae. मल्टीसेलुलर साइटोप्लाज्म के साथ हाइपी रहता है, उसको कोइनोसाइटिक हाइपी कहा जाता है. ओदर्स हैव सेप्टेट और क्रॉस वॉल्स. दिस क्रॉस वॉल्स इन देयर हाइपी. द सेल वॉल्स ऑफ फंजी आर composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from dead substrates and hence are called saprophytes. Those that depend on living plants and animals are called parasites. They can also live as symbionts in association with algae as lichens and with roots of higher plants as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spores called conidia or conidia or sporangiospores or zoospores, and sexual reproduction is by oospores, ascospores and basidiospores. The various spores are produced in distinct structures called fruiting bodies. The sexual cycle involves the following 3 steps. What are the steps? Fusion of protoplasms between two motile or non-motile gametes called plasmogamy. Fusion of two nuclei called karyogamy. Meiosis in zygote resulting in haploid spores. Then, when a fungus reproduces sexually, two haploid hyphae of compatible mating types come together and fuse. In some fungi, the fusion of two haploid cells immediately results in diploid cells. However, in other fungi, Ascomycetes and Basidiomycetes, and intervening dikaryotic stages n plus n, that is 2 nucleic per cell, occurs. Such a condition is called dikaryon, and the phase is called dikaryo phase of fungus. Later, the parental nuclei fuse and the cells become diploid. The fungi form fruiting bodies in which reduction division occurs, leading to formation of haploid spores. The morphology of the mycelium, mode of spore formation and fruiting bodies forms the basis for the division of kingdom into various classes. Now we'll study about Phycomycetes. Members of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places or as obligate parasite on plants. The mycelium is aseptate and coinocytic. Asexual reproduction takes place by zoospores or by एप्लानोस्पोर्स. जूस्पोर्स मोटाइल होता है, एप्लानोस्पोर्स नॉन मोटाइल. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphology, isogamous or dissimilar, anisogamous or oogamous. Some common examples are mucor, rhizopus, the bread mold mentioned earlier, and albigo, the parasitic fungi on mustard. Then we will study about Ascomycetes, commonly known as sac fungi. The Ascomycetes are mostly multicellular, that is penicillium, rarely, or unicellular, that is yeast, Saccharomycetes. They are saprophytic decomposers, parasitic or coprophilous, growing on dung. Mycelium is branched and septate. The asexual spores are conidia, produced exogenously on the special mycelium called conidiophores. Conidia on germination produced mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like asci, singular ascos. These asci are arranged in different types of fruiting bodies called ascoscarps. Some examples are Aspergillus, claviceps, and Neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and buffels are edible and are considered delicacies. Then morels and buffels, edible. Then Basidiomycetes. Commonly known forms of Basidiomycetes are mushrooms, bracket fungi, or bubbles. They grow in soil, on logs and tree stumps, and in living plant bodies as parasites, that is rust and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotype. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis take place in the basidium producing 4 basidiospores. The basidiospores are exogenously produced on the basidia. The basidia are arranged in fruiting bodies called basidiocarps. Some common members of agaricus mushroom, Australagos moth, and Poxinia fungus. Deuteromycetes, commonly known as imperfect fungi. It is called as imperfect fungi because only the asexual or vegetative phases of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stages have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect sexual stages of members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual spores known as conidia. The mycelium is septate and branched. Some membranes are saprophytes or parasites, while a large number of them are decomposers, that is, litter and help in mineral cycling. Some examples are Alternaria, Colitotrichum, and Trichoderma. Then we will see about kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organisms, commonly called plants. A few members are partially heterotrophic, such as insectivorous plants or parasites. Bladderwort and Venus flytrap are examples of insectivorous plants, and Cuscuta is a parasite. The plant cells have an eukaryotic structure with prominent chloroplast and cell wall mainly made of cellulose. You will study the eukaryotic cell structure in detail. Plant includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Life cycle of plants have two distinct phases: the diploid saprophytic and haploid gametophytic, that alternate with each other. The length of the haploid and diploid phases and whether these phases are free, living, or dependent on others vary among different groups in plants. This phenomenon is called alteration of generation. You will study further in details. Then we will study about kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cells lack cell walls. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserves as glycogen or fat. Their mode of nutrition is holozoic. By ingestion of food, they follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanisms. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Then virus, virids, and lichens will study. In the Five Kingdom classification of Whitaker, there is no mention of some acellular organisms like virus and virids and lichens. These are briefly introduced here. All of us who have suffered the ill effects of common cold or flu know what effects viruses can have on us, even if we don't associate it with our condition. Viruses did not find a place in classification since they are not truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate. cell to replicate themselves, killing the host. Would you call virus living or non-living? The name virus, that means venom or poisonous fluid, was given by Pastor D.J. Ivanovsky in 1892. Recognized certain microbes as causal organism of the mosaic disease of tobacco. These were found to be smaller than bacteria because they passed to bacteria proof filters. M.W. Bizernik demonstrated that the extract of the infected plants of tobacco could cause infection in healthy plants and called the fluid as contagium vivum fluidum. Contagium vivum fluidum, infectious living fluid. W. M. Stanley, 1935, showed that viruses could be crystallized, and crystals consist largely of proteins. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to proteins, viruses also contain genetic material that would be either RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single stranded RNA, and viruses that infect animals have either single or double stranded RNA or double stranded DNA. Bacterial viruses or bacteriophages, viruses that infect the bacteria, are usually double stranded DNA viruses. The protein coat called capsid, made of small subunits called capsomeres, protects the nucleic acid. These capsomeres are arranged in helical or polyhedral geometric forms. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by a virus. In plants, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing and stunted growth. Viroids. In 1971, T.O. Diner discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA.